Thanks very much indeed. It's a great honour to accept this honorary doctorate on my own behalf, on behalf of all the great people who've come through my lab and done the great work that I'll be telling you a little bit about in my talk. I'd like to thank the faculty here for nominating me for this award. So I'm just going to start off with this picture here. You'll be familiar with this. It's the sensitive plant, mimosa. The sensitive plant, and of course the thing that irritates me about calling this plant the sensitive plant, if you can't guess, is it gives the impression that mimosa is the only sensitive plant, but actually all plants are sensitive plants. So they have to be sensitive because otherwise they get wiped out by diseases. And there's a lot of very important plant diseases. And this is from an issue of a page in Science magazine from a few years back. Wheat stem rust, potato blight, I'll say more about that. This is a parasitic plant, Asian soybean rust, viruses, rice blast, black sigatoka disease of banana. These are very, of course, enormous losses worldwide. And we just heard a lovely talk from Ed Cahoon about the wonderful products that plants already make and that they can be improved to make. We don't want to have that lost to disease. So plants have an immune system, and it's always good to reflect on evolutionary history to try and make sense of things. And a very important concept is that single-cell organisms have very different disease problems than multicellular organisms. So, because if you've got multicellular organisms have a lot of spaces between the cells into which parasites and pests can inject themselves to compete for the nutrients. And plants and animals evolved multicellularity independently. So the origins of the plant immune system must be way back here. This was then when plants and animals diverged before, in fact, the appearance of multicellularity in eukaryotes. So what we know is that then resistance mechanisms must have evolved independently in plants and animals, and although we now know that plants use some similar protein domains and mechanisms in their innate immunity. Mammals have this unique phenomenon of acquired immunity with antibodies and so on. So what do we know about plants and their disease resistance mechanisms? So I think the key insights really came from genetics. And here are some of the key geneticists. This is Biffin, who didn't really come up with a wheat ear this big, and the others are named here. But Biffin showed about 100 years ago, or slightly more than 100 years ago, soon after the rediscovery of Mendel's work, that disease resistance to wheat rusts was inherited as a dominant Mendelian trait. I need to do this so that this is visually recorded. And then that's an important discovery. But this guy Steakman made another important discovery, which is that not all rusts are the same. So there are races of wheat rust that can be resisted by a particular resistance gene in wheat, and there are other races that can overcome the resistance that's in that wheat. And so there's strain specificity in whether or not a particular strain of the rust is resisted, and that imposed part of the discipline of plant pathology is keeping your strains pure and not mixed up. That was a new technique that had to be developed. And then Floor and Ort independently showed that if they analyzed the properties of races that could overcome a resistance gene, they did genetics on the pathogen, and I then looked at the genes that conditioned the capacity to overcome a resistance gene, those genes are recessive. In other words, the pathogen had to stop doing something that it did in order to overcome a resistance gene. And so virulence is recessive. So on that platform now, we can actually think quite broadly and address these kinds of questions about plant disease and plant disease resistance. And so we can analyze why different wheat accessions vary in their resistance to wheat rust. What's the genetic basis? We can now understand the genes for many of condition many of these variations in resistance. We can also ask questions that are broader. Why is wheat resistant to barley powdery mildew? And why is barley resistant to wheat powdery mildew? This is sort of non-host resistance. And of course, if we could understand the genetics of that, that might open up a new source of resistance genes we could use in agriculture. And then a very different kind of question is, why is wheat resistant to very non-adapted pathogens such as potato late blight? And if we could understand these immune receptors well enough and how they work, that opens the way to moving them from one plant species to another and opening up the whole of plant diversity as a source of disease resistance for our crop plants. And then we're also interested in the mechanistic basis of how plant immune receptors work, and I'll say a little bit about that towards the end. So key concepts in plant immunity are as follows. So first of all, unlike mammals, plants don't have cells that move around in the body and attack invaders. Every cell is stuck in its little cardboard box and it can only defend itself locally. So innate immunity in plants, heritable immunity, is cell autonomous, is a language that we use. So plants have got really good defense mechanisms. We don't need to add any new defense mechanisms. They make reactive oxygen species, they make antibiotics, they undergo programmed cell death that restricts the spread of pathogens that depend on living host cells. But for this to work, the plant has to know when the pathogen shows up. Defense activation depends on perception. And this perception depends on receptors, cell surface or intracellular receptors, protein molecules whose job it is to detect when pathogens show up or detect molecules from pathogens and use that as a trigger to activate defense mechanisms. And this is a sort of cartoon that illustrates that. So here are your cell surface receptors. They'll have often receiving domains, leucine-rich repeats, or other domains, and then protein kinase domains inside. They recognize so-called pathogen-associated molecular patterns, or PAMPs. And this is pretty good at activating defense, and then you get this PTI response that, I'm failing to control my arrow here, that can stop the pathogen growing. So for pathogens to overcome this, they have to make molecules that suppress plant immunity, and these are called effectors. They can either be delivered from bacteria by type 3 secretion or by unknown mechanisms still from these projections that go into host cells from co-adapted pathogens like rusts and mildews. It's called a haustorium. But these, in turn, these effector molecules whose job it is to suppress host immunity can actually also trigger host immunity if there's an immune receptor within the cell called a MBLRR or NLR receptor in the cell, and that can activate defense in turn. And if you have a recognized effector, that's called an avirulence gene, and that's what you have a mutation in that Floor showed you need to mutate to overcome a resistance gene. So there's now, because it was a puzzle always, why on earth would pathogens carry molecules that enable plants to detect them? It seems like a really stupid thing to do, right? But the reason is they make a big population of effector molecules, any of which the plant could evolve to detect and activate defense in response to, and any particular one where the plant has evolved that, mutations in that recognized effector become virulence genes that enable the pathogen to overcome the resistance gene. So now our knowledge of these receptors is actually very useful. So this just illustrates some work I did with my then postdoc, now independent investigator Cyril Zipfel, with the Arabidopsis cell surface receptor, recognizes a conserved bacterial molecule. The real point is this: the perception capacity of different plant families is not identical. So some plant families can recognize molecules from pathogens that other plant families just can't recognize. So this Arabidopsis, and present in all the Brassicaceae receptor called EFR, recognizes bacterial EFTU, and Solanaceae, like tomato and potato, do not have this perception capacity, but by simply moving the receptor from one plant to another, in this case from Arabidopsis to tomato, you can convert something that's susceptible to Ralstonia bacterial wilt to something that's resistant to bacterial wilt. And this is a very useful thing to do. So I talked about these intracellular receptors, these NLRs, stands for nucleotide binding leucine-rich repeat receptors, amongst other things. I'll spare you all the jargon. And it comes in two flavors. And plants and animals both have these types of receptors, or though the animal, in the animal one, the nucleotide binding domain part is slightly distinct from what it is in plants. In plants, you've got the so-called embark domain, and in animals, it's called NACH domains. All these little letters have to do with homology that's detected in one protein or another. So, but typically, plants have hundreds of these NLR encoding genes. And animals, well, mammals don't have very many. So humans have 20 or so, mice 30. But there are some classes of animals, such as sea urchins, sea anemones, corals, which have hundreds. So how do these kinds of receptors work? So we take an interest in these questions. How do they evolve and function in populations? What are their roles in immunity to non-pathogens? And how do they convert recognition of pathogens into defense activation? So when you're thinking about resistance gene function, or NLR protein function, they've evolved in plant populations that are highly heterogeneous and diverse. And of course, what we've done in agriculture is to generate plant populations where you have millions of genetically identical plants. And if you've got millions of genetically identical plants, and one of them is unfortunate enough to get a disease, then you know this isn't going to end well. So the colors here are meant to indicate perception capacities, or resist NLR types, or R genes. So if you've got a plant that's got one type of R gene, and you've got a pathogen that overcomes it, then you're going to have a field of dead plants. And this is the risk we run when we plant agricultural monocultures. Over here, what these different colors are meant to signify is, let's say, alleles of a resistance gene that recognize there's 5 different colors, so 5 different molecules from the pathogen. So then if you get a pathogen comes in, then it's got a long way to go for the epidemic before it can find another susceptible, because it can only grow on red gene encoding plants, whereas all the other colors are resistant to it. So that's intuitively, obviously, going to slow down an epidemic, and there are other benefits as well I don't have time to go into. And of course, that's what agriculture used to be like at the time of Vincent van Gogh. And what you can see here is not, of course, you can't see the genetic heterogeneity in this field, but you can also see, there's a lot of other plants in this field, which is part of what also slows down epidemics. So for resistance gene to work, you need diversity in your populations of hosts, and to adapt our knowledge of resistance genes to use them to control crop diseases better, we need to operate with awareness of that context. Now, I've told you about this idea that you can move receptors from one plant to another. So what I'm going to tell you about our work is really kicked off by this paradigmatic paper from the Staskovitz lab. They've been interested for a long time in resistance to a bacterial disease, Anthomonas, in pepper and tomato, and they cloned, after a huge labor, a gene from pepper called BS2, which is one of these NLR encoding genes, and it confers resistance to a relatively indispensable effector from Xanthomonas, so it's a very good resistance gene. In tomato, there's basically no genetic variation at the disposal of breeders for resistance to this disease, and in southern Florida, it's a huge problem. So what Brian showed was that if you take the pepper BS2 gene and put it into tomato, then you've got a resistant tomato, and if you haven't got the resistance gene in, then you've got a dead tomato. And this is the tomato. This is the gene, the plant that the resistance gene came from, pepper. The BS2 gene confers resistance there too, and if you don't have it, then you've got a dead pepper. And so this is a gene that people have been eating for generations when they eat peppers that come from southern Florida. But if you put it into tomato, suddenly, of course, because of the neurosis about GM, it becomes a problem. Nevertheless, as a scientist, you have to have the courage of your convictions. And we have been looking at genes for resistance to various diseases from wild relatives, that could be useful in crops. So one of the systems that we chose to work with is this pathogen, which I'm sure is going to be a very important pathogen of cardamine, Ed Cahoon. So I think that the resistance genes we've isolated against it may prove useful. It's called Albugo candida. It's a white rust. It's an oomycete pathogen, and basically there are a number of races, each of which is specialized on different hosts. And so we're interested in the differences between these races and understanding the basis for host specialization. And we think this is important. You get these eruptions of dangerous angry white pustules, and there's been outbreaks in the United States recently. We had a nasty outbreak in the UK. And there was risk of outbreaks on the continent as well, and I hope we'll all agree resistance to angry white pustules is very important. So what resources of resistance are there that we can draw on? Arabidopsis, it turns out, is quite a useful source of resistance to brassica infecting races of white rust. So we use genetic tricks to dig into the NLR gene repertoires of 19 different Arabidopsis accessions, all of which are resistant to brassica infecting albugo races. The key trick is to identify a susceptible. We use transgressive segregation. We screen for lines that were susceptible. Because you imagine, if you're trying to work on non-host resistance and everybody's resistant, then what do you cross to what to do genetics on it? You can't get anywhere until you've actually identified a susceptible. So we found, our hypothesis was that different Arabidopsis accessions would all be resistant because of NLR genes, but it'd be different repertoires in each. And if you cross and self, you're going to get segregants that lack any of the NLR genes in either parent that confer resistance, and that's what we found. So then we could do genetics on it, and we chased it down and I'll spare you the details, 5 different NLR encoding genes of this tier NLR class. And so what we can then do is to move these into Brassica rapa, or Junsia in this case, and here's one of the genes we cloned, and we actually get useful resistance to white rust in this crop, which is an important crop of particularly of oilseed crop for India, where there's considerable losses to this disease. So specialization then of these races involves evolving to evade detection by NLR immune receptors. So we've understood why Arabidopsis is resistant to this brassica infecting race. The race that can grow on brassica can evade the brassica repertoire of NLRs. So if we can move the NLR repertoire from Arabidopsis, once we found the right ones, into brassicas, then we've got a good way of conferring disease resistance. A much more important crop and a much more important disease is potatoes affected by potato late blight. So what we'd really love to do is to be able to turn potato into a non-host for this disease, Phytophthora infestans. Turn it into a plant on which Phytophthora infestans simply can't grow. And there's various groups around the world undertaking this kind of work. We've turned to this particular accession, Solanum americanum, a worldwide diploid, self-compatible relative of potato, moderate genome size, a lot of genetic diversity, and we're trying to clone as many R genes as possible from this, resistance to Phytophthora infestans from Solanum americanum. That's what RPI AMR genes means. We found at least 5, not cloned all of them yet, but we think if we can stack all of these into one potato genotype, using the kind of modular cloning where we put a bunch of genes on the same tDNA that Ed referred to earlier, then we will generate a genotype of potato that'll be very hard for the Phytophthora to evolve to overcome. And we've done some experiments on this in the field. Our first experiment with this field experiment with RPI VNT1 from Solanum venturii. And now we've cloned multiple more additional NLR genes, and whoops, there we go. That's what I was looking for. So this is a picture taken exactly a month ago or so, and this is my colleagues and I in a field trial we ran this year, where we took RPI AMR1E, RPI AMR3, RPI-VNT1, 3 different R genes, and also a stack of all 3 in the same plant. And basically you can see what happened to the plants that didn't get any RPI genes. They are toast, and the individual R genes all worked, and so we're pretty confident the stack of 3 at once will confer quite durable resistance to the pathogen. So then the question is, so a more basic science question, which may be useful in the future, is how do these things work? And so our studies on basic mechanisms of resistance genes we use an Arabidopsis-sourced pair of resistance genes called RPS4 and RS1 for resistance to Pseudomonas syringae 4 and Ralstonia solanacearum 1. And this is a pair of genes that work together to achieve recognition of a couple of bacterial effectors called AVR-RPS4 and POP-P2. Gravity, in case you're wondering why the arrow moves when my hand isn't on it. So these have the canonical type structure. We've got tier domain, embark domain, Lucian repeats, and various other domains down here. And most notably, you've got down here a Werke transcription factor domain, which we now know to be involved in the detection. So we've got essentially an input-output problem here. We know that the tier domain of RPS4 up here is the domain that actually does the signaling upon recognition. So we've got somehow or other a chain of events that convert interaction with the effector down here into output up here. So what do we know about this? We know that R4 on its own can constitutively activate defense. We know that if we co-express R1, it can block that. So this is negatively regulating the whole thing. We know that the tier domains of R4 and R1 actually can form a complex with each other. We have a co-crystal structure of that. And we have evidence that that interaction is relieved upon defense activation, perhaps freeing up this to go off. We actually have data that the Werke transcription factor domain down here is negatively regulating the complex via its interactions with this domain, domain 4. And what we think happens is that the effector somehow or other relieves that negative regulation of the Werke domain on the whole complex. And once that's been relieved, then this interacts with a domain here, and that kicks off further action up the chain. But there's still a lot to figure out at the protein domain mechanism level for us to figure out how this works, and also the roles of other genetically defined players with these acronyms here. So there's a big basic science program going on in my lab on this question, and I think I don't need to show you a lot of Western blots to say this is quite a tough problem, and we're making as much progress as we can. But here's a sort of a conceptual model of what we've now believe, and this is work of our lab and also a French lab, Laurent Delon's lab. So these are the 2 effectors. Now, I've told you this sort of paradox. Why on earth would pathogens make molecules that enable them to be detected? Well, AVR-RPS4 and POP-P2, the POP-P2 is an acetyltransferase that acetylates Werke transcription factors, and AVR-RPS4 interacts with them. These transcription factors are utterly crucial to plant immunity. And so if you've got effectors that interact with them, then it's likely this is going to result in a more susceptible plant. And so that's why if your pathogen's got effectors that can mess with the action of Werke transcription factors, it'll be good at imposing susceptibility on the host. So what's happened in evolution is that the plant has somehow evolved a Werke domain containing immune receptor. So the plant needs to know when something's messing with its Werkes. Okay, so it's got a immune receptor complex that carries a Werke transcription factor domain, and then when the AVR-RPS4 and POP-P2 attack these to impose susceptibility, it can't help alerting the plant to the fact that it's something's showed up that's messing with Werkes, and that results in defense via this recognition complex. And this is such a good idea, you'd have thought plants would have thought of it more than once, and indeed they have. So I've told you about this pair here, which have a Werke transcription factor domain at the end of RS1 that participates in an immune receptor complex with RPS4. And then there are rice genes, RGA5 and RGA4, that are involved in rice blast resistance. And they have a different domain called an HMA, heavy metal associated domain down here. And they recognize effectors from rice blast that interact with HMA proteins. again to promote virulence from the standpoint of the pathogen. So this led to this concept called the integrated decoy hypothesis. And in fact, if you look at plant genome databases from like 35 plants or so, this is the work led by my colleague Ksenia Krasileva. She surveyed the NLR encoding genes from many plant species for the presence of domains that are other than the canonical domains in NLRs. And this is what she found displayed as a word map. So you see protein kinase domains, you see worky transcription factors, you see thioredoxins, you see HMA domain proteins, and a number of other domains. And these domains overlap with the repertoire of domains that are identified when you search for plant proteins that interact with pathogen effectors using methods like yeast-2 hybrid. So this is a quite satisfying explanation for the presence of these types of receptors. I don't want you to get the impression that all plant NLRs carry integrated domains or integrated decoys, but in the Graminea, for example, between 5 and 10% of them do. So it's obviously been a device that has been deployed frequently in plant evolution to help plants detect pathogens and activate defense accordingly. So here's what I've tried to tell you. So all plants are sensitive plants, not just the mimosa. And NLR proteins contribute to this sensitivity that enables them to detect molecules from pathogens that get inside the plant cell, and they can use this as a cue to activate defense. And these kinds of proteins are involved in both plant and animal innate immunity. There's tremendous diversity in plant NLR repertoires in plant populations, and we can tap into this when we look for new sources of resistance that we want to deploy in our crop plants. And the NLRs can contribute to, quotes, non-host resistance, in other words, why Arabidopsis is resistant to brassica infecting races of albugo, not just to Arabidopsis infecting races. And these can be used, this extension of the repertoire we can draw on can be useful for crop improvement. These integrated domains in NLR proteins are instructive when you think about host-parasite coevolution. And I haven't gone into detail about this, but we can use sequence capture methods to accelerate our investigation of resistance gene or NLR encoding gene diversity, and also of pathogen diversity. And we wrote a review on this recently, if anybody's more interested in this topic, that came out in Science at the end of last year. And I'd like to thank again those who invited me for this honor. And I'd also like to thank my wonderful colleagues, collaborators, and funders, particularly the Gatsby Foundation, which has funded my work at the Sainsbury Lab over the last 29 years. Thank you very much.